Zaraz wyłączę to światło.
Dzień dobry.
Dzień dobry.
Dzień dobry.
Dzień dobry, dzień dobry. Spróbuję włączyć prezentację. Nie wiem, czy widać teraz?
Widzimy wszystkie slajdy teraz.
O, to dobrze. To już jesteśmy, połowa sukcesu jest osiągnięta. Patrzę, gdzie my żeśmy ostatnio skończyli. Mi się wydaje, że gdzieś tutaj. Okej. Okay, so during previous lecture, we discussed the structure of helicases, and you remember that they are combined in the multimeric enzymatic machineries, and usually these are hexamers or heptamers, and sometimes even it could be dodecameric structure, and this is just example of archaea, and the helicase isolated from archaea, which forms the heptameric ring. However, regardless of if this is heptameric or hexameric, the monomer structures are very similar. So protomer structures, which combines the multimeric assembly of a helicase, are similar, and this one is from E. coli, and this one is from Methanobacterium thermoautotrophicum. So this is just the conclusion of the structural analysis. I also mentioned that when we look at the eukaryotic helicases, so in contrast to prokaryotic or archaea helicases, all protomers in prokaryotic systems and archaea systems, or protomers combining the hexameric or heptameric structure of a helicase are identical. But in eukaryotic systems, we cannot say about the individual protomers combining hexameric structure, but we have to say about the individual proteins, which are called MCMs, so mini chromosome maintenance proteins, MCM2, MCM3, MCM4, MCM5, MCM6, and MCM7, and all of them have different size, different lengths of polypeptide chains. You see that it varies from even more than 1,000 amino acids to something which is 700 amino acids in length. And of course, there is at least at the core similarity. However, the N-terminal ends and C-terminal ends are different in length and in the amino acid compositions. And this is the concept that some of those subunits have a regulatory function, and some of them have catalytic function, and some of them may have also the structural function. But no matter what, we have to just discuss how it works, how the helicase works. And the first issue which bothers scientists for years was, okay, we have heptameric, hexameric, or dodecameric rings. So how it is possible that actually the DNA, how it's attached and how it enters to this internal channel, which is inside this multimeric structure. So there were a few concepts, and we will discuss this briefly. So one was just ring opening. So we may have ring, however, it could be somehow open, and you can consider that most likely accessory protein, which we discuss as DNAC or others, can open the structure. So that was the concept. The other is a treating, so that would be kind of bizarre concept, but then we need to have the end of DNA to introduce into the channel, or the DNA scaffold has to be cutted in one place just to anneal the helicase onto double-stranded or single-stranded DNA. So this is the second concept, and the first concept is just the disassembly of the helicase and reassembly on the DNA. So we can easily imagine that the helicase protomers could reform the complex on the DNA, and that would make the placing of double-stranded DNA inside the channel. Okay, so we have 1, 2, 3 concepts. The question is which one is true, or maybe not. One is true, but a few of them are possible in cells. And the proposed model, the first proposed model, is that there are some subunits of a helicase which are involved in this opening of the ring, and the specific size of the helicase, which are marked here in green and in red. And in red are going to be those sites which are inside the channel, and a site place for the interaction with DNA and placing the DNA inside the channels. And that was the paper published in Proceedings National Academy of Science in 2012, showing for the first time that the helicase from archaeal bacteria could form the open ring structure. And you see that this is just a reconstitution of this structure. And you see that the helicase has this open kind of a channel, and these are individual subunits of the helicase. And there is a gap in between 5 and 2. And when you look at the electron micrographs, so I don't know, maybe in here or in here, or also in here, you can see that indeed the structure is somehow open. And this is just the model. And that was just the proposition that it depends on the ATP concentration in the solution. So that regarding if there is an ATP present, okay, or ADP, so the present inside the ring, that may cause opening or closing of ring structure. And it is believed that the DNAC protein actually is important for this ring opening, However, DNA-I, which we discussed, so this is just the homologue of DNA, most likely is responsible for the reassembling of the helicase, de novo assembling of helicase quaternary structures, so these multimeric structures on the DNA. So that still these two possibilities are relevant, and we have to consider that the helicase ring could be open, but also the helicase could be reassembled onto the double-stranded DNA and single-stranded DNA de novo from individual protomers. And it's going to be a little bit different mechanism, but resulting with the same ending product. Okay, in here. Okay, so this is how we can kind of assume how the helicase could be loaded on the DNA. But the other story is how the helicase performed the unwinding of DNA. And that was just very many hypotheses. I just don't want, maybe it's not worth to go deeply in those ancient hypotheses, like a rolling circle, that the helicase is a passive or active mechanism. So it was just the concept that the helicase has a passive mechanism for the catalyzed DNA unwinding. So it would be like a DNA is breathing, so associate and dissociate the double-stranded structure. And at that moment, when there is a dissociation of double helix, then helicase moved and blocked the reassociation. And we know that this is not true, that the mechanism for the is active mechanism. Also, that was an active mechanism for the dimeric helicases, so that there is a dimer of a helicase, which actually kind of rolls on the DNA, and this is ATP-dependent reaction. And when it rolls, particular subunit of this dimeric helicase it caused the ATP hydrolysis and unwinding of a DNA. Okay, and so the other model is just DNA exclusion model, and that was proposed for T7 gene product 4 helicase, and this interaction with unwinding junction of 30 base pairs of duplex DNA and 25 base pairs of single-stranded DNA was presented, and the end is docked in a central channel. 3' end is excluded. So actually this is 5' end and 3' end, and the helicase actually stays on the single-stranded DNA and moves. And so how it moves, it's another story. And we can have a wedge model, torsion model, and helicase destabilizing binding model. So, so different concepts were considered as a mechanism for the, but you see that they are not that specific. Okay, so what is the current model? So the current model says that there is a sequential change of interaction with nucleotides of particular subunits within the multimeric structure of a helicase. And this is related with ATP binding, hydrolysis, and release of ADP. And it's in the sequential manner it happens. So that results with conformational changes, which results with rotation or movement of a helicase on the DNA, or we can say otherwise that the helicase translocates DNA inside this channel. Okay, so now I will go to the details. We may have 3-side sequential model, 2-side sequential model, on 1-side sequential model. it is believed that most likely free-side sequential model is the true. So we will have the gray circles are those subunits of a helicase which are structural subunits, so non-catalytic subunits, and the color marked in the green, red, and blue are those helicase subunits which are catalytic ones. So if we have let's look at this position. So when look at this, so the green subunit is bound by ADP. Am I right? Maybe I'm confused myself. Okay, so this is just empty. Okay, this is just, this is ATP and ADP interacting subunits, okay? And within the time, you will see what happens. That the blue subunit, which is empty, and this, let's say this is time 1, and this is time 2, and this is time 3. So it's like sequential what happens within the subunits. So the empty subunit has a T, so it's bound by ATP. The subunit, the green one, which interact at this moment, ADP, release the ADP, and it's empty. And this, which had the ATP because of hydrolysis, has an ADP right now, okay? And then in the next time, so this sequential what happens, the empty one, so this one, okay, now is loaded with ATP. ATP comes here. This one, which had an ADP, releases ATP, and it's empty right now. And this one, which was bound by ATP, so this one, because of hydrolysis, is bound by ADP in the time 3. So you see that this is just the sequential what happens with individual subunit, which is a catalytic subunit. If it's bound by ATP, at the next step, phosphate is released, would be bound by ADP, and in the next step, because of releasing ADP, it would be empty. Okay, so this is sequential event of each individual subunits, and at the same time, it's impossible that all of them are empty. One would be empty, one would be bound by ATP, and the second one would be bound by ADP. So this is what happens, and this is just the concept, and you can see, so this is exactly what happens in time, and you see that the helicase can kind of rotate on the DNA template. And when we look at the red subunit only, so, and this is just, of course, the simplifying picture of the subunit. So we can see that the subunit interacts DNA and it's bound with ATP. And in the next step, because of a hydrolysis, it changed the conformation. It's bound by ADP, sorry, because of a hydrolysis. And you see that it somehow rotates the DNA or rotates itself on the DNA template. Yes, then with the empty state, the DNA is released, the interaction with release with this one subunit. And after interaction with ATP, the DNA binding is de novo established by this particular subunit. So if when one subunit bounds DNA, the other could be dissociated from the DNA interaction. And the third one, at the same moment, would bound DNA, but in the different side. Okay? And that makes the possibility of rotation of DNA and rotation of the helicase with a DNA channel. Rotation of DNA with a DNA helicase channel inside the channel. Okay, and we have to remember that talking with eukaryotic helicases, that would be absolutely the same, but those protomers, those subunits are structurally different. So it looks like that the ATP hydrolysis drives this situation, and now I'm just going to show you the similar enzyme actually doing completely different. It's going to be ATP synthase, and there is some similarities, or I would say maybe not similarities, but it would show you how two enzymes can catalytically could be similar, however, would perform completely different work. So what is the connection? Okay, so the ATP synthase is an enzyme which actually is important for the ATP synthesis. And ATP synthesis is based on the proton and electron transport during phosphorylated oxidation and during the respiratory chain. So what happened in each cells or mitochondria or bacteria cells, the proton gradient is formed due to transportation of protons outside the membranes. Okay. So protons are pumped from the matrix into the inner membrane space, and total ton protons and translocate per pair of electrons move from NADH to oxygen. And this results with proton gradient. And then ATP synthase activity is driven to create ATP from ADP and phosphate. And this activity is driven by the movement of protons inside the channel because of this gradient which is formed here. Okay, so actually protons are pumped outside in there and then are retranslocate inside the cell or mitochondrium through this ATP synthase through this channel of this enzyme. So actually this is this channel, and this is this more generalized structure of a channel. And it was found that due to this proton movement, this subunit of ATP synthase rotates. That makes this gamma complex in epsilon, also rotation, and then they change the conformation of alpha subunit, which are in here, and at the end, so this is F1, and then you will see that that caused the synthesis of ATP. Okay, it was shown by the attaching these rotations were visualized by actin filament attachment on this gamma complex in here and attachment by his sacs, the beta complexes of ATP synthase onto the lead. And this rotation of the filament through the fluorescent was observed. And this is just the original picture from nature showing this rotation of a filament on the top of an enzyme, of course, artificially attached. You see this. And I don't know if that would work. I will try to show you the animation. So we have protons, proton movement, then ATP synthesis from ADP and phosphate group. And those are those subunits. You see the channel. Okay, this is just ATP synthesis place for synthesis. Alpha and beta subunits on the top. Rotation. This is top view of a beta subunit, so like catalytic ones. Alpha is not presented in this picture. You see ADP and phosphate, rotation of gamma, conformational change of all three subunits, the other conformational change, movement of gamma and conformational change, ATP hydrolysis, and ATP release. Yes, and once again. So, just, we have stage reaction 1, 2, 3, 4. We have empty, bound by ADP. The ADP changing of the conformation and ATP synthesis, and then would be released. So we have open structure, bound by ADP, bound by ATP, ATP released, ATP present, and rotations of subunits. When you think about this, it's absolutely very similar comparing the helicase. However, there is no proton movement which drives the enzyme, but this is the ATP hydrolysis which changes the conformation of catalytic subunits. And because of a changing of a conformation, then something happens inside the enzyme, the ring, which makes the helicase movement, the DNA movement inside, or the helicase movement on the DNA template. Okay, so this is just this the comparison showing the two enzymes actually doing completely different work, but the ATP hydrolysis could be the fuel for the energy source for the conformational changes, or conformational changes could be generated by the proton movement, and that would result with the ATP synthesis. So this is just the comparison of those two. Okay, so I think we will stop this part. Okay. Come on, what's it? And we will move to the second part, but before this, I will go. Okay, can you see this, the new slide?
Yes, we can.
New presentation?
Yes.
Yes? Okay, so let's do 15 minutes break and then we will continue on eukaryotic helicases and eukaryotic replication origins and origin recognition complexes, okay? Yes, 15 minutes break.
Okay.
Yeah. Mm-hmm.
Sorry, we cannot hear you.
Hear nothing.
Sorry, but we can't hear you.
You are mute.
We can't hear you.
I think you have to unmute yourself.
This unmute button is right on the right when you have your camera. On Teams application.
Musi pan otworzyć aplikację Teamsów i koło przycisku zakończ będzie.
O, właśnie tutaj.
Teraz mnie słyszycie?
Tak?
Problem nie polega na tym, że ja nie wiem, gdzie to zrobić. Problem polega na tym, że mam raz, dwa, trzy screeny i w ogóle nie mogłem się do tego dostać. To niestety tak jest czasami. Także w ogóle nie mogłem tego okna znaleźć, dlatego musiałem zamknąć aplikację. Nie? No dobrze. Teraz nagle widzę siebie jakoś w jakiejś czarnej otoczce. Też tak mnie widzicie?
Widzimy pana w jakimś pokoju.
Tak? No dobrze. To teraz przynajmniej się widzimy i słyszymy. Teraz jeszcze muszę uruchomić tą prezentację. Tylko nie wiem, czy ją widzicie. Pewnie nie.
No właśnie, tak trochę jest to problemem, bo takie, o, teraz jest w ogóle lepiej.
A teraz?
O, teraz widzimy tylko pana.
Ja nie mogę. Co się tutaj stało? Bo ja teraz uwspólniam screen. O, teraz może to zadziała.
No teraz widać pana pulpit. I tak, teraz widać też już prezentację.
Dobrze, może się uda, może się uda. Teraz muszę kursor przesunąć na ten ekran. Udało się. OK, chyba jest dobrze. So, back to replicon model. In eukaryotic systems, we have more replicators, more replication initiation proteins, and more initiation sites at the chromosomal DNA, on the chromosomal DNA. And so we may have more than one chromosome and more than one replication initiation site, so more origins, so more replication forks. And at bacterium, we have one replicon, and it's going to be millions of base pairs length. But in eukaryotic systems, we may have even thousands of replicon, and the length of them are pretty short. So it could be even 300 bases in length. So multiple replication initiation events and multiple replication sites are identified. We may consider simple origins or metazoan origins, and it depends on the structure, but no matter what, it's going to be origin recognition element, DNA unwinding element, A plus T reach site, and this is just the core of the origin, and it's bound with origin recognition complex, so replication initiation proteins, not the protein by proteins, which form the complex at origin recognition element, and the entire origin has also auxiliary elements, and auxiliary elements are bound by transcription factors. And replication proteins as origin recognition complex bound the core elements of replication origin. And you see that it could be very complex, but nevertheless, we have auxiliary, two element, origin, AT, and core, and different factors can interact the auxiliary part of replication origin. Yes, this is just a simplified example of interaction of T antigen with origin structural elements, and T antigen has a hexameric structure. It's a replication initiator, but the same, also the helicase. It has a helicase activity, so very bizarre structure, which the bizarre protein, which at the same time is an initiator and a helicase, but you see that it can interact the auxiliary element and the core element of replication origin. And this is just this T antigen simian virus electron microscopy showing the structure, and you see that it looks a little bit like a helicase with particular subunits with a channel inside, but the side view is a little bit different. It's like this, and that can form the double hexameric structure, one hexamer and the other hexamer, and these are inverted repeats within the replication origin, and auxiliary elements and AT reach part of the replication. In yeast, we have specific complexes which are called autonomous replication sequences. And these autonomous replication sequences contains autonomous replication sequences consensus sequence. Okay, so this is just the consensus sequence which is in here, which is important to be a part of autonomously replication sequence. So that would be that the autonomously replication sequence contains consensus, which is important for the recognition by proteins. So some sequence specificity is there. And when you look at the modular architecture in yeast of this autonomous replication sequences, you see that there is a particular parts, an A part, B part, B part, and B part. And when you look at the plasmid stability, which actually can identify the what is required, what structure is required for the activity of this replication origin, you see that the A part is absolutely essential because there is no replication activity. However, some replication and the deletion of some parts like B, deletion of B or deletion of D is giving some activity, replication activity, but it's very diminished. It was found that the origin complex interacts, so ORC, subunits, ORC1, ORC2, ORC3, ORC4, ORC5 subunits, interact this autonomously consensus sequence. And also some of those subunits interact the B, so this not essential part, but important part of replication origins. This is in Saccharomyces cerevisiae. In Saccharomyces pombe, you see that there is a specific ORC4 proteins form specific hook, which actually interacts with AT-rich part of a replication origin, and that brings the entire complex interactions with the DNA, which was tested by the DNase1 footprinting in here. And again, the A part is the most important for the interaction. So A-rich DNA. And this is just the Drosophila heron gene and the amplification enhancement region, and something which is called ACE, amplification control elements, which were found important from Drosophila melanogaster origin activity. So this is organization of a third chromosomal Horion gene. And it was found that this is just the ORI-B origin element, which is ACE and the amplification enhancer region. And this is just simplified model of this Drosophila melanogaster ORC interactions with ORI-B and ACE3 elements. So you see that simplifier models are simplifier models, well, no matter what. There is some sequence requirements, and regardless if we are looking at the Saccharomyces system or Drosophila, so higher organisms, we see some similarities, some sequence specificities of the DNA to origin recognition complex interaction. So how it is in humans, and that was the very long debate. Finally, somehow stopped and let's say characterized in 1998 that the paper published in Science magazine demonstrated that in humans we also have specific sites for the replication initiation, and it was done by closing particular fragment of a human DNA into simian system cells and identified if this particular part of a DNA can serve as a site for DNA replication. It was beta, the site which contains the gene for the beta globin, and the specific experiment with bromodesoxyuridine labeling. So that's supposed to be bromodesoxyuridine labeling. So that was a pulse of bromodesoxyuridine, which was incorporated into the DNA. Then that was just the isolation of these newly synthesized fragments by specific antibodies and testing what gene was actually synthesized, so where the DNA replication started. And this is just was done by PCR, and it was identified that the replication was just conducted within the beta globin gene, and this is just the PCR testing of this experiment. And so that tells that also in humans we have particular sites which serve as a beginning of a replication synthesis, so actually replication origins. However, it is still not clear what actually makes these sites specific. And it is believed that this is not exactly the sequence, but is something which we call the initiation zone, which contains usually potential DNA unwinding element, and it consists origin of bidirectional replication, but different post-transcriptional modifications, modifications of DNA can also, like methylation, can have an effect if this origin is active or not active. So this is more complex, and it seems like not necessarily sequence matters, but also how the DNA is condensed, if it's methylated or not methylated, and that matters about the replication, origin, activation at particular moment, and activation of particular DNA unwinding elements. So those unwinding elements very many of them could be present, but not very many of them could be activated at the same time, at the same moment. Okay, so this is just about the regulation and the state on the DNA at the metazoan cells. Okay, so this is just the concept that the chromatin structure, nucleosome positioning, methylation of DNA, superhelicity of a DNA matters for the origin in eukaryotic cells and higher eukaryotes. And that makes the specificity of replication origin in eukaryotic. Okay, so this is just the conclusion. You can read later on about this, so that there is very many replicators and occupied by origin recognition complexes. More replicators are used than are needed to complete S phase. Replicators fire asynchronously through the S phase. Replicator usage, so origin replication usage, can be regulated during development and during different metabolic conditions. And once per cell, genome duplication does not require specific replicator sequences. And different replicators have different DNA sequence requirements. Some have no DNA sequence requirements. And many different features of chromatin structure and function can influence the activity of replicators. And initiators, so replication initiator proteins are origin recognition complex, are homologous and are found in all eukaryotes. And origin recognition complex occupies replicators in vivo, and the factors that influence DNA origin activity of origin recognition complexes are hardly divergent. Okay, so that was proposed, and it's still accepted, this concept that we have to replace the concept of a replicator as a specific DNA sequence with that of a context-depending element that is recognized by the initiator, so origin recognition complex, through several features which can include DNA sequence. So what is origin recognition structure? So I mentioned those bizarre drawings, and we of course understand that this is not necessarily how it looks like. This is just the structure of origin CDC's origin recognition complex part, CDC6 origin protein. And we mentioned before that it contains AAA plus domain, okay, with ATP, and wing helix domain, and this is typical for origin recognition complex proteins. And this is just phylogenetic tree, ORG4, ORG3, ORG2, ORG5, CDC6, ORG1. So you see that they are in some related, and the phylogeny of those proteins are kind of complex. But there is one important thing, that when they form, this is just a paper, Nature paper published in 2015, when the origin complex from Drosophila melanogaster was reconstituted. So all proteins consisting the origin recognition complex, ORG1, ORG2, ORG3, ORG4, ORG5, and ORG6 were purified, and the complex was assembled. And what you see here, these are protomers of those origin recognition complex proteins. And this is just on panel A, the structure of them. So you see that all of them have AAA plus, AAA plus, AAA plus, AAA plus, AAA plus, and they have wing helix domains as well. But when you look at the complex, they just form the kind of like a ring with a central channel inside. So we can expect the DNA could be placed in here. And then when you look at the side view of this, so, and you have to compare this with this one. So this is this violet color, and we see AAA plus domain of ORG4, and then is a hinge, and then we have triple wing helix of ORG4, but you see that wing helix of ORG4 interacts AAA plus domain of ORG5, and then there is a hinge, and wing helix ORG5 interacts AAA plus ORG5, and AAA plus domain, there's a hinge, and wing helix ORG3 interacts AAA plus of ORG2. Yes, So this is one protein, this is one protein, and then there is another protein. So it's like a domain sharing. So one protein gives the hand to the other protein. ORC4 gives the hand, so the hand is Wink-Hellis domain, to ORC5, and ORC5 gives a hand to ORC3, and ORC3 gives a hand, so Wink-Hellis domain, to ORC2, and then consists the structure. Okay. These ORC proteins are kind of like a hybrid of both clamp loader and prokaryotic DNA protein, and these are superpositions showing these similarities. And all of them, like DNA A and also replication factor C, so clamp loader, and contains this ESMs. Okay, so initiator specific motifs, and those motifs are usually directed into the inner space of this channel, which was shown in here. Okay, so these motifs are directed to the channel, and of course those are important for interaction of with the DNA. What we have in here, so this is ORC AAA+ domain is disengaged from ORC4 and sits above the plane. Okay, so that maybe we'll see it better on the next slide. Okay, so what it shows you that in this structure, this ISMs are in here, so DNA is over there. But when you see there is some kind of a structural rearrangement over there and over there that makes this structure opened. So you can imagine, and this opening is based on this two positions, or maybe it's easily to see over there. So when you look, the ring is not closed because of this and because of that. So these are positions of ORC1 which are not in plane. So the ring in here is not closed. You see this? And you saw also, you can see it over there. And then this makes the possibility of loading of origin recognition complex containing ORC1, 2, 3, 4, etc. onto the DNA, because those subunits are not in plane and the ring is not closed. Okay. Yeah, so we kind of come from this picture to this one, that one, and then more simplified, this one. Okay, I think that we can stop in here, and the next lecture will talk about the helicase complex in eukaryotic system. So how the MCM complex looks like. Maybe you have some questions. Or you would like to talk again about some aspects. I don't hear. So thank you very much, and we will see next week, and we'll talk about the MCM complexes and activities of those complexes. Thank you very much, and bye-bye.
Goodbye.
Thank you. Goodbye.
Goodbye.
Thank you. Goodbye.
Goodbye.
Goodbye. Goodbye.
you.